Hello future doctors. Welcome back to Conquer Pathology. I'm Dr. P and today we are going to conquer genetics. Now when you open Robbins, this chapter looks terrifying. It's full of enzymes, mutations and complex diagrams. But here's the secret. Genetics is just math and logic wrapped in biology. Today we are going to crack the code. By the end of this hour, you will not only understand the difference between autosomal dominant and recessive, but you will also look at a pedigree chart and solve it in under 30 seconds. We will cover the vocabulary of genetics, Mendelian disorders, the big four, the ultimate pedigree cheat sheet, my personal algorithm, non-Mendelian patterns, the big three, and we'll conclude the lecture with studying the clinical integration, connecting pathology to pediatrics and medicine. Let's dive in. Part 1. The vocabulary of genetics. The building blocks. Before we build the house, let's look at the bricks. You need to know these four terms because examiners love them. Penetrance versus expressivity. Penetrance. This is binary. It's a yes-no question. Does the person with the mutation show the disease? Imagine a light switch. If the switch is on, you have the disease. If you have the gene but no disease, that is incomplete penetrance. Example, BRCA1 gene. Some women have the gene but never get breast cancer. Expressivity. This is a volume dial. You have the disease, but how bad is it? The music is playing, penetrance is yes, but is it a whisper or a rock concert? Example, neurofibromatosis type 1. One patient might just have cafe au lait spots, which are mild, while their sibling has massive neurofibromas and scoliosis, the severe form. This is variable expressivity. Pleiotropy versus genetic heterogeneity. Pleiotropy. One gene messes up many systems. Example, Marfan syndrome. The fibrillin defect. It affects the eye, lens dislocation, bones, tall stature, and heart, aortic dissection. One stone, many birds. Genetic heterogeneity. Many different genes cause one phenotype. Example, deafness. You can have a mutation in any of 50 different genes, but the result is the same, hearing loss. The blueprint and the typos. Think of the human body as a skyscraper. The genome is the entire set of blueprints for the building. Chromosomes are the massive folders containing different sections, plumbing, electrical, structural. Genes are the specific instructions for one single thing. Example, the faucet in room 302. Codons are the words in the instruction. Example, use brass bolt. Mutations are a typo in that instruction. Types of instruction errors or mutations. Point mutation, swapping one letter. Example, use brass bolt becomes use glass bolt. The bolt is there, but it's weak. This is sickle cell anemia. Glutamic acid becomes valine. Frame shift mutation, deleting or adding a letter. Example, instructions, the big red dog, delete the B, then it becomes the IGR EDD OG. The entire instruction becomes gibberish. Example is Duchenne muscle dystrophy. Trinucleotide repeats or stuttering. Example is do not 100 times, then enter. Example is Fragile X syndrome. Before we move on any further, let's learn how to draw a pedigree chart. In pathology and medicine, a picture is worth 1,000 words. But a pedigree chart, that's worth an entire diagnosis. Today, let's learn how to draw a perfect pedigree in 3 simple steps. Step 1. The symbols. First, you need to know your shapes. It's simple geometry. The male, draw a square. For female, draw a circle. If the sex is unspecified, draw a diamond. The patient or the proband is the person sitting in front of you. You have to draw an arrow pointing to their symbol, bottom left corner. If they are the first person in the family to get sick, they are the proband. If they are just the person asking for advice, they are the consultant. Step 2. The connections. For marriage or mating, draw a horizontal line connecting a circle and a square. For consanguinity, if they are relatives like cousins, draw a double line. This is a huge red flag for autosomal recessive diseases. For offsprings or sibship line, draw a vertical line down from the marriage line, then a horizontal bracket. Draw siblings in birth order from left to right, oldest to youngest. Step 3. The status. Now let's tell the story. Who is sick? Draw the affected one by shading it completely black. Leave the unaffected one empty or white. The carrier in autosomal diseases is drawn half shaded or vertically split. The carrier in X-linked diseases is drawn as a dot in the center of a circle. The deceased subject is drawn as a diagonal slash through the symbol. A deceased subject is drawn as a diagonal slash through the symbol. Here is exactly how you draw it when the examiner is watching. Start with the proband, draw the arrow and their symbol, square or circle. Go up, draw the parents above. A deceased subject is drawn as a diagonal slash through the symbol. Here is exactly how you draw it when the examiner is watching. Start with the proband, draw the arrow and their symbol, square or circle. Go up, draw the parents above, label this generation 1 on the left. Go down or across for siblings. Draw the proband's brothers and sisters. Label this generation 2. Number them. Number individuals from left to right in each generation using Arabic numerals like 1, 2, 3. Example, the proband might be 2, 3, generation 2, third person. Shade. Fill in the affected people last so you don't make a mistake. And that's it, a clear professional pedigree. Always add a key or legend in the corner of your page explaining your symbols. Examiners love that. Go practice drawing your own family tree. It's the best way to learn. And now let's move on to the next part. Part 2. Mendelian Inheritance, the Big Four. Now let's talk about single gene disorders. In Robbins, these are divided into 3 buckets: autosomal dominant, autosomal recessive, X-linked, recessive and dominant.
Let's break them down using my rule of thumb system.
Autosomal dominant. The loud personality, you only need one bad copy to show the disease. Autosomal dominant disorders usually involve structural proteins or receptors, not enzymes. Why? Because if you are building a wall or structure and 50% of your bricks are broken, the wall falls. Transmission is vertical. You see it in every generation, grandpa to mom to child. Key examples are Marfan syndrome, defect in fibrillin 1, familial hypercholesterolemia, defect in LDL receptor, achondroplasia, FGFR3. Huntington's disease, it is a trinucleotide repeat. Let's integrate this with pediatrics. Since these are structural, the onset is often later in life or young adulthood, like Huntington's, unlike autosomal recessive disorders which present early. Autosomal recessive. The shy personality. You need 2 bad copies to show the disease. Parents are usually carriers, heterozygous, and healthy. Autosomal recessive disorders usually involve enzymes. Why? Because enzymes are efficient. If you have 50% functional enzymes, you are the carrier. Usually that's enough to do the job. You only get sick if you have 0% enzymes. That means you need to be in a homozygous state. It is transmitted horizontally. It skips generation. Suddenly, 3 siblings in one generation have it, but the parents are fine. The red flag is consanguinity, marriage between relatives. The key examples here are, let's have a look at my mnemonic for autosomal recessive disorders. A, B, C, D, E. Albino, beta thalassemia, cystic fibrosis, deafness, which is sensory neural, and enzyme deficiencies, like in storage diseases. X-linked recessive diseases. The mother passes it to the son. Males are affected because they have XY chromosome. Females are carriers. Transmission. Knight's move pattern. It skips a generation, grandpa to carrier mom to a deceased grandson. The crucial rule here is no male-to-male transmission takes place. A father gives his Y to his son. He cannot give an X-linked disease to his son. The key examples are hemophilia A and B, G6PD deficiency, Duchenne muscular dystrophy, and color blindness. X-linked dominant, the all daughters rule. One bad X is enough for anyone. X-linked dominant does not skip generations. It looks like autosomal dominant, but with one key difference. The dead giveaway is an affected father will pass the disease to all of his daughters and none of his sons. Let's integrate this vertically with dermatology. Incontinentia pigmenti is a classic example. It's often lethal in males. They die in utero, so you only see affected females. Wow, that's a lot. Now the question arises, is there any sequential questionnaire for pedigree studying as to know what kind of genetic disease it is, or is it any mitochondrial disease? To conquer any pedigree in your exams, you don't need to guess. You just need to ask the family tree 5 specific questions in a fixed order. This is Dr. P's sequential pedigree protocol to reach the diagnosis every single time. Step 1. The generation gap test. The question you need to ask here is, does the disease appear in every single generation, or does it skip generations? If it appears in every generation, think dominant and go to step 2. If it skips generation, think recessive and go to step 3. Step 2. The dominant branch. If you decided it is dominant, ask this question. Does an affected father pass it to all of his daughters and none of his sons? If the answer is yes, the diagnosis is X-linked dominant disease. Logic here is, he gives his only X, the affected X, to daughters and his clean Y to the sons. If the answer is no, then he passes it to a son. The diagnosis is autosomal dominant disease. The logic here is, male-to-male transmission is the gold standard proof that it is not X-linked. Step 3. The recessive branch. If you decided in your step 1 that it is recessive, then ask this question. Are mostly males affected, and is it passed from a healthy carrier mother? If the answer is yes, diagnosis is X-linked recessive. Logic here is, females have a backup X, males don't. If the answer is no, then males and females are roughly equal. The diagnosis is autosomal recessive. Look for consanguinity, the double lines between parents. Moving on to step 4, the maternal only test. This is a special case. You have to ask the question, does an affected mother pass it to all of her children, sons and daughters, while an affected father passes it to none? If the answer is yes, the diagnosis is mitochondrial inheritance. The logic here is, mitochondria comes only from the egg, the oocyte, never the sperm. Example is Leber's hereditary optic neuropathy. Step 5. The Y-linked test. This is the rarest. Question is only males affected, and does every affected father pass it to every son? If the answer is yes, diagnosis is Y-linked, Hollandric. The logic here is, only men have Y. Example is hypertrichosis spinae or hairy ears. This is the summary decision table or the cheat sheet. If you see father to son transmission, it cannot be X-linked, must be autosomal. If all daughters of affected father are affected, likely X-linked dominant. All children of affected mother are affected, likely mitochondrial. Skips generation, but mostly males, likely X-linked recessive. Skips generations, an equal male-female ratio, likely autosomal recessive. If you are stuck between autosomal dominant and X-linked dominant, look at the affected father's daughters. If even one daughter is healthy, it is autosomal. If all daughters are sick, it is X-linked. We are going to play medical detective. To master Robbins Chapter 6 Genetics, you must be able to look at a family tree or the pedigree and diagnose the inheritance pattern instantly. We will walk through 2 classic clinical scenarios, hemophilia A and Marfan syndrome. Case study 1. The bleeding boy, hemophilia A. Let's look at the clinical scenario. A 4-year-old boy, Rohan, is brought to the clinic with massive swelling in his right knee after a minor stumble. His mother mentions that her father, Rohan's Nanaji, died young due to a bleeding disorder, and her brother, Rohan's Mamaji, also has frequent joint swellings. Let's map this out using standard symbols. Squares as males, circles as females, shaded as affected, and dot in the center as carrier. The disease skipped Rohan's mother. She is healthy, but her father and her son are affected. Let's make our first observation. The disease skipped Rohan's mother. She is healthy, but her father and her sons are affected. So the rule was, if it skips a generation, it is recessive. The second observation would be, only males are affected in this family. So the rule was, if there is a heavy male bias, think X-linked. Observation 3, the clincher. Rohan's father is perfectly healthy. And the rule was, in X-linked recessive, the bad X comes from the mother. So this is an example of X-linked recessive inheritance. Let's integrate this with biochemistry. Mutation in the F8 gene causes deficiency of coagulation factor 8. In pathology, you'll see a prolonged APTT, intrinsic pathway defect, but a normal PT and bleeding time. So, moving on to medicine, the treatment involves factor VIII replacement. Never give aspirin to these patients. Case 2. The tall teenager, Marfan syndrome. Let's look at the clinical scenario. Aditi, a 16-year-old girl, stands 6.1 feet tall. She has very long fingers, arachnodactyly. Her father is also very tall and recently underwent surgery for an aortic aneurysm. Her younger brother is starting to show similar features. Let's draw the pedigree. Now let's make few observations. Observation 1. There is no skipping. Aditi has it, her father has it, and her brother has it. The rule says vertical transmission means dominant disease. Observation 2. Both males, father and brother, and females, Aditi, are affected. The rule says if there is no sex bias, it is autosomal. Observation 3. The risk for the next child is exactly 50%. So it all boils down to autosomal dominant disease. Let's integrate this with anatomy or histology. There is defect in fibrillin 1, which is a scaffold for elastin. Look for cystic medial necrosis in the aorta. Integrating it with surgery, the biggest threat is aortic dissection. In ophthalmology, look for ectopia lentis, which is upward dislocation of the lens. Remember the Steinberg sign, the thumb sign, and Walker-Murdoch sign, the wrist sign for your clinical exams. Here's the cheat sheet for your exam comparing hemophilia A, which is an X-linked recessive disease, and Marfan syndrome, which is an autosomal dominant disease. So till now we have mastered the rules of genetics: dominant, recessive, X-linked. It was neat, organized, and logical. But biology is messy. Today we are talking about non-Mendelian disorders. These are the rebels. They don't follow the standard rules. If you see a pedigree that looks impossible, like a healthy dad having 3 sick kids with a dominant disease, or a disease getting worse with every generation, you are dealing with one of these. We are going to cover the big 3 non-Mendelian patterns listed in the NMC competencies: trinucleotide repeat mutations, the stuttering genes; mitochondrial inheritance, the maternal power; and genomic imprinting, the parent trap. Let's crack the code. Part 1. Trinucleotide repeat mutations, the stutter. First up, trinucleotide repeats. Imagine you are typing a sentence and suddenly your keyboard gets stuck. You want to type cat, but you type cat, 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 cat. This is what happens in your DNA. A specific 3 nucleotide sequence expands massively. Anticipation. The disease appears at a younger age and with more severity in recent generations. Let's look at this scenario. Grandpa got tremors at 60, dad got tremors at 40, the son gets tremors at 20. Why? Because the stutter gets longer and more unstable as it is passed down. The big 3 diseases. Must know for exams. Huntington's disease, autosomal dominant. The defect is in CAG repeat on chromosome 4. Let's look at the mnemonic. CAG, caudate loses acetylcholine and GABA. Clinical integration with medicine and neurology. There is chorea or dancing movements, dementia, and depression. The expansion happens during spermatogenesis, which is paternal expansion. So if the dad passes it, the anticipation is worse. Next one is Fragile X syndrome. It is X-linked dominant. The defect is CGG repeats in the FMR1 gene. Mnemonic is CGG. Chin is protruding, giant gonads, or macroorchidism. Let's integrate this with pediatrics. This is the number 1 inherited cause of intellectual disability. The expansion happens during oogenesis or maternal expansion. Now the third one is myotonic dystrophy, autosomal dominant. The defect is CTG repeats. Mnemonic is CTG, cataracts, toupee or balding, and gonadal atrophy. The clinical sign here is myotonia, difficulty relaxing muscles. Example, can't let go of a handshake. Part 2. Mitochondrial inheritance, the maternal power. Next, let's talk about the powerhouse of the cell. Here's the golden rule. Sperm do not contribute mitochondria. They are too busy swimming. They leave their batteries or mitochondria in their tail, and the tail falls off during fertilization. Result, all your mitochondria comes from your mother. The cheat code for pedigrees. As you know, if you see a chart where affected mother passes it to all her children, the sons and daughters. Affected father passes it to none of his children. The diagnosis is mitochondrial inheritance. Let's look at this concept, heteroplasmy. If mom passes it to everyone, why is the son severely affected but the daughter only mildly affected? This is called heteroplasmy. Imagine a bag of marbles. These marbles are mitochondria. Some are red or mutated, some are blue or healthy. When the mother's cell divides to make the egg, the marbles are split randomly. Egg A might get 90% red marbles or severe disease, and egg B might get 10% red marbles, mild to no disease. This explains the variable expression. Key examples here is Leber hereditary optic neuropathy, which causes sudden blindness in young adults. Part 3. Genomic imprinting. The parent trap. This is the trickiest concept of all, but I'll make it simple. Usually we have 2 active copies of every gene, one from mom and one from dad. But in imprinting, one parent's copy is silenced or turned off by methylation. It's like putting tape over the mouth of that gene. Paternal imprinting, the dad's allele is silenced, you only rely on mom's. Maternal imprinting, the mom's allele is silenced, you only rely on dad's. The classic exam question is chromosome 15, Q11, Q13 deletion. The same deletion causes 2 totally different diseases depending on whose chromosome is broken. Let's see with an example of Prader-Willi versus Angelman showdown. The Prader-Willi syndrome. The problem is paternal deletion. Dad's copy is deleted or missing. Normally, the Prader-Willi gene is only active on dad's chromosome. Mom's is naturally silent. If dad is deleted, you have 0 active copies. The mnemonic is Prader or paternal deletion. Symptoms are hypotonia at birth, then hyperphagia, uncontrollable eating, causing obesity and hypogonadism. Angelman syndrome. The problem here is maternal deletion. Mom's copy is deleted or missing. Normally, the Angelman gene UBE3A is only active on mom's chromosome. If mom's is deleted, you have 0 active copies. The mnemonic is mom's an angel. Maternal equals to Angelman. The symptoms are happy puppet syndrome, inappropriate laughter, ataxia, seizures, severe intellectual disability. Part 4. Gonadal mosaicism, the hidden assassin. Last one. This is a rare curveball. Imagine phenotypically healthy parents have one child with an autosomal dominant disease like osteogenesis imperfecta. You think, oh, it must be a new mutation. Then they have a second child with the same disease. You panic, wait. The odds of 2 new mutations are 1 in a billion. What is happening? Here's the explanation. The mutation is not in the parent's blood or somatic cells. It is only in their gonads, the sperm or eggs. A patch of cells in the dad's testes or mom's ovaries has the mutation. So they keep churning out mutated gametes even though the parents themselves are fine. The cheat code here is healthy parents plus multiple affected siblings with a dominant disorder. It could be gonadal mosaicism. Part 5. The non-Mendelian pedigree algorithm. Let's update our pedigree algorithm from part 1. Start looking at the parents of the affected child. If mother is affected, does she pass it to every single child? Does the affected father pass it to no one? If the answer is yes, then it is mitochondrial disease. If inheritance looks dominant or vertical transmission but gets worse or presents earlier in life in subsequent generations. If the answer is yes, then it is trinucleotide repeat or anticipation. If parents are healthy but multiple kids have a dominant disease, if the answer is yes, then it is gonadal mosaicism. Before we finish, let's link this to your other subjects. Biochemistry. Mitochondrial DNA. It is circular, double-stranded, no histones. It codes for electron transport chain enzymes for oxidative phosphorylation. This is why mitochondrial diseases causes lactic acidosis where aerobic respiration fails. Pediatrics. Fragile X. Look for the child with delayed milestones, autism spectrum features, and later the large ears or testes. Prader-Willi syndrome. The floppy baby who becomes the obese child. This is a classic spot diagnosis. Obsgyne. Hydatidiform moles. This is an imprinting problem too. Complete mole. There are 2 paternal sets, 46XX, all dad, no fetus. Ovarian teratoma. There are 2 maternal sets, 46XX, all mom, no placenta. Lesson. You need both mom and dad's imprinting to make a viable baby. That's it. We have now covered the rebels of genetics. Let's summarize this. The stuttering repeats cause anticipation, Huntington, Fragile X. Mitochondrial is maternal only, Leber's. Imprinting depends on who gave you the gene, Prader-Willi or Angelman. So that's it. I have a homework for you. Go open Robbins, look at pedigrees and try to solve them without reading the caption. So far, we have looked at the words in the book of life, the single genes or Mendelian disorders, and the tricky sentences, non-Mendelian disorders. Now we are zooming out. We aren't looking at typos in a sentence anymore. We are looking at missing chapters, torn pages, and books that have been glued together. This is cytogenetics, the study of chromosomes. And we are going to cover the diagnostic tools you will use in real life to find these errors. Karyotyping, FISH, PCR. By the end of this segment, you will know exactly which test to order and why. Let's open the library. Chromosomal basics. First, some definitions for your Viva. The chromosome structure. Imagine a chromosome as an X. The center knot is the centromere. P arm is the petite arm, the short arm. Think P, petite. Q arm is the long arm, it follows P. G bending, we stain them with GIMSA. This creates light and dark bands. Dark bands are gene poor or heterochromatin. Light bands are gene rich or euchromatin. The normal count. Euploid. The exact multiple of n, which is 23 in humans. Humans are diploid. 2n equals to 46. Aneuploid, not an exact multiple. It could be 45 or 47. This is an error. Numerical abnormalities, the counting error. How do we end up with 47 chromosomes? The culprit is usually non-disjunction. Mechanism. Imagine a couple, that is chromosome pair, going through a divorce, that is meiosis. They are supposed to split up and go to separate houses, the daughter cells. Normally, one goes left, one goes right. But in non-disjunction, one partner clings to the other. Both go to the same house. The other house gets nothing. The result is one cell has 24 chromosomes, trisomy when fertilized, and one has 22, monosomy. The big three trisomies. You need to know these three by heart. Down syndrome. It occurs in chromosome 21. The mnemonic is D for down, D for drinking. 21 is the legal drinking age. Key features here are flat facies, simian crease, ventricular septal defect, atrial septal defect, endocardial cushion defect, Alzheimer's, more than 40 years of age, Edwards syndrome. It is trisomy 18. The mnemonic is E for Edwards, E for election. You vote at 18. The key features are rocker bottom feet, clenched fist, overlapping fingers, and micrognathia. Patau syndrome. It is trisomy 13. Mnemonic is P for Patau, P for puberty. Puberty hits at 13 years of age. The key features are P for Patau, P for palate, the cleft palate, polydactyly, and holoprosencephaly. Dr. P's cheat code. Look at the numbers. 21 is the smallest of these chromosomes, 13 is the largest. Trisomy 21, survival is common. Trisomy 13, these are most severe defects. Death usually occurs before 1 year of age. The bigger the extra chromosome, the more lethal the imbalance. Next part is structural abnormalities or the torn pages. Sometimes the count is 46, but the structure is broken. This is harder to catch. First one here is translocations or swapping seats. Reciprocal translocation, 2 chromosomes swap pieces. No genetic material is lost. Usually the patient is normal, but their gametes, sperm or egg, will be unbalanced. This leads to recurrent miscarriages. Robertsonian translocation, it happens only with ectocentric chromosomes like 13, 14, 15, 21 and 22. These have very short P arms. 2 chromosomes fuse at the centromere. The 2 long arms join to make one giant chromosome. The 2 short arms fall off and are lost. Result is the count is 45, but functionally normal because P arms of ectocentrics are just junk DNA anyway. The danger here is if a parent has a Robertsonian translocation involving 21, they can pass extra 21 material to the child, causing familial Down syndrome. Deletions. The missing page. Cri-du-Chat syndrome. Deletion of short arm of chromosome 5. The baby cries like a cat. because of the laryngeal defect. Isochromosome, the mirror image. The centromere divides horizontally instead of vertically. You get a chromosome with 2 P arms or 2 Q arms. The classic example here is Turner syndrome variant, isochromosome XQ. Next part is sex chromosome disorders. Nature tolerates sex chromosome errors better than autosomal ones. Why? Because of lionization or X inactivation. The first one here is Turner syndrome, 45X. The rule is the only whole monosomy compatible with life is Turner syndrome. You cannot survive missing an autosome. Clinically, you see following features. I remember this by the clown's mnemonic. Coarctation of aorta, lymphedema, ovaries are streaked, webbed neck, nipples are widely spaced, and short stature. Next is Klinefelter syndrome, 47XXY. It is male hypogonadism. It is usually due to maternal non-disjunction. Mom gives XX and dad gives just Y. Clinically, you see tall stature, gynecomastia, small firm testes, and infertility. Hormonal studies indicate high FSH and LH, testes aren't working, low testosterone, and high estrogen. Next, we'll see the toolkit, genetic diagnostic techniques. This is the most practical part. A patient walks in, which test do you order? Let's dive in. First one is karyotyping, the map. What it is? We freeze cells in metaphase, stain them, and take a picture. It is best for big problems like aneuploidy, Down syndrome, and large translocations. The limit of it is, resolution is low. It's like looking at a city from a satellite. You can see if a building is missing, but you can't read the street signs. It is a slow procedure. It takes around 2 to 3 weeks. Next one is FISH, or fluorescence in situ hybridization. It is the searchlight. What is it? We use a fluorescent DNA probe that sticks to a specific gene. Go fish for a specific sequence, we say. It is best for specific questions. Does this patient have the DeGeorge deletion 22q11? Too small for karyotyping? Does this leukemia patient have BCR ABL translocation 922? The pros for this test are, it is fast. It takes around 24 to 48 hours to give the results. It doesn't need dividing cells. It can do so in interphase also. The cons are, you must know what you're looking for. You can't just say, check everything. The third one here is the chromosomal microarray, the scanner. What is it? It is a chip. We scan the whole genome for gains, the duplications, or losses, the deletions. It is best for when you say, I don't know what's wrong. Child with multiple congenital anomalies and normal karyotype. It is the first line test for intellectual disability and autism. The next one is PCR, or polymerase chain reaction, the photocopier. It amplifies a tiny piece of DNA millions of times. It is best for single gene mutations like trinucleotide repeats, sickle cell. It's like finding a needle in a haystack by making the needle grow into a sword. Dr. P ordering algorithm. If it is an obvious syndrome, like Down's feature, order karyotype to confirm and check for translocation. If a specific microdeletion is suspected like DeGeorge syndrome, go for FISH. If there are intellectual disabilities and multiple anomalies, the cause is unknown, go for microarray. And if it is single gene defect, like in cystic fibrosis, go for PCR or genomic sequencing. Alright, let's wrap this up. The key takeaways here are, Robertsonian translocation carriers have 45 chromosomes but are normal. Their kids are at risk for Down syndrome. Turner is 45X, the only viable monosomy. Klinefelter is 47XXY, the extra X comes from non-disjunction. FISH is for specific targets and microarray is for scanning. Now let's give you a homework. Look up bar body. How many bar bodies does a Klinefelter patient have? How many does a Turner patient have? Hint, the rule is n-1. Put your answer in the comments. I've created a high-yield PDF cheat sheet of all these tables and flowcharts. The link is in the description. If you enjoyed this detailed breakdown, hit that like button and subscribe to Conquer Pathology with Dr. P. Let's crush those exams together.